This is a lecture that would have been given on Wednesday, April 8th. It covers Block 3, Unit 8, Clinical Trials. So as usual, we'll go through concerns and business items. We'll review what's happening in the lab, which, as you probably guessed, is not a lot. We'll cover also material that we've already talked about, and this will help you hopefully continue to develop the way you talk about your research to others and also in your final research paper. And then we'll review briefly drug discovery and development. But first, let's go over some upcoming due dates. Last Sunday, you would have completed the Block 3, Unit 8 Clinical Trials Play Posit. The grades overall were very, very good, so congratulations. The research paper introduction draft would have been submitted on Sunday, and I think there weren't any late submissions, which is fantastic. I want to encourage you all to reach out to me and Maureen if you have any questions on the next draft, the methods draft, which is due this coming Sunday, April 12th at 10 p.m. The introduction draft, I know speaking only for the Monday, Wednesday sections, I heard from very few groups, and I think for the methods section especially, this is the place where you really want to reach out because we can help you develop that section so you get a good draft and you get really meaningful feedback. The idea with submitting these drafts is that you will get the feedback, it'll be graded, but it's pretty low stakes grading. And then what you'll do is you'll incorporate the suggestions made by the person who reviews your drafts and both me and Maureen. You'll incorporate those into your final paper. So now that you've submitted your introduction draft, for example, you'll get the edits back, make those changes, and you have a piece of your paper already done. And the same would be true for your methods section. The third exam is coming up soon. That's going to be on Wednesday, April 15th. So that's 2 weeks from when you'll probably listen to this lecture on April 8th. It's going to be administered online again. And we're going to make sure that it gets opened on time and that you have 50 minutes to complete it. We did extend the time for the Block 2 exam to make sure everybody had enough time. If you had any major concerns with Block 2 or suggestions on how to improve the format of the exam, we're all ears and are willing to take any suggestions that you have. The week and after your midterm exam, on Sunday, April 19th, you have the data analysis exercise. And as we've mentioned before, the data analysis exercise is intended to take the place of the labs that we don't have anymore. So what we've done, and I'll explain it in just a minute in more detail, is developed 4 tutorial videos that will walk you through how to analyze a compound library. And you'll be reporting the results of your own compound library that you tested earlier in the semester as part of this data analysis exercise. The exercise will be posted on Wednesday, April 8th, and it's due, as I said, on Sunday, April 19th at 10 p.m. This is an individual submission, so do not submit it with your groups. Everyone should submit their own assignment. You'll be analyzing the data from your own library screen. So this is from the screen that you performed earlier in the semester. And we'll be walking you through questions that will help you refine the data and also report any hits and graph those hits appropriately, with the idea that this is going to help you report the sort of hypothetical data that you get related to your own independent project in your final paper. So what's happening in the lab? Nothing. There's nothing happening in the lab. We've been cleaning it up. But you should know that before Sydney and Maureen and I sort of backed off of campus, we quantified survival of all of the vials that were remaining. So we do have a complete library for you to analyze for the data analysis set. You're probably wondering what happened to the flies. All of the flies have been autoclaved. We held back about 40 bottles, I think, and those will be used by independent study students over the summer, hopefully, and if not, in the fall, we'll maintain those. Because the play posit for this unit, for the clinical trials unit, went so well, I thought maybe what we could do is take some time and do a more thorough review of what we've already covered. I think overall the exam average was higher than it was last time, so that's really good news. A lot of people did very, very well. If you have any concerns about your grades, do reach out to me and Maureen, and we can give you an explanation and hopefully some tips on how to better prepare for the next exam, because as I said, the format is going to be the same as it was for exam 2. So let's do a pretty thorough review. Starting with what cancer is. So when you're developing your final research paper, if you look at published manuscripts, you'll see that the first thing that these papers do in the introduction section is tell you about what the problem is. Because a lot of people in labs are studying things that on the surface might not appear to be that big of a problem. Now, you're fortunate because you're studying a problem that most people are familiar with. So you're having to sell your project, but not as heavily as someone who's studying a tiny protein that's expressed in a cell that nobody's heard of. So what you'll want to do in your final research paper, and I'm sure you've already done this in your introduction, is introduce the idea of cancer. And although you're writing specifically for people who are pretty familiar with the subject, it's always good to demonstrate that you understand what the problem is. The first way that you can do that is to describe what cancer is. And I know it seems awfully silly to have to do that in a manuscript, but if you look really carefully at manuscripts that are published, like I said, you're going to see that you're really being hit over the head with what the problem is. Cancer arises from mutations, and it's typically thought that a cancer will come from a single cell that has one mutation that confers some ability to the cell that it didn't have before. Those abilities can be described in the hallmarks of cancer that we'll see in the next slide. But over time, generally, that one mutation is going to increase the rate at which that cell divides, or its ability to divide in an environment that normally would be quite resistant to cell growth. So those cells divide and divide and divide, and over time, they develop more and more and more mutations. And the way that I think about this isn't the way that you're going to write about it in your research paper, but the way that I think about it is developing superpowers. You have a superpower that allows you to get really, really big. You have a superpower that allows you to produce more of yourself. And then all of a sudden, you develop, totally coincidentally, the power to break out of the tissue that you're existing in and travel to a new tissue. And that's called metastasis. That's when you start developing, or a cancer patient would start developing tumors in other areas of their bodies. And this is a point where the patient's cancer becomes really difficult to treat, because each of these tumor sites are going to contain cells that have fairly unique mutation composition within them. So a single treatment is very unlikely to be able to kill all of the cells in these 3 different tumor sites. The hallmarks of cancer are a set of criteria that were developed to describe what's happening inside of a tumor or in a cancer cell. Cancers don't express all of these generally, and there isn't even any specific order in which these are developed inside a cancer cell. So evading growth suppressors might occur first, or perhaps sustaining proliferative signaling would occur first. Maybe resisting cell death would occur first. But generally, you can think of cancer as having one or more of these hallmarks, which makes those cells special because they're not responding to the normal signals that allow growth when it's appropriate and kill the cell when it's appropriate. So currently, there are a lot of strategies for treating cancer. We have radiation and many, many different types of chemotherapies, but generally those chemotherapies are very toxic, so they're really difficult for the patients to tolerate, and they're not very successful. There are only a handful of usually blood cancers that are cured by things like chemotherapy and radiation or stem cell transplants. So there's a great need for identification of new drugs. And those drugs currently are identified through high throughput screens. So these high throughput screens are much higher throughput than what we're doing. You all tested, I think, around 340 compounds this semester. When we talk about high throughput at the level of a pharmaceutical company or a large lab, we're talking about tens of thousands of compounds in a very short period of time. Another strategy is targeted development. So through knowledge of the cancer gained through genomics or understanding the genes that are expressed, proteomics, the proteins that are produced in the cells that are cancerous, and bioinformatics, which uses all of that data together to try to put the pieces of the story together. You can pick targets, and in a fashion that's similar to a high throughput screen, you start to look specifically for compounds that will inhibit or affect the function of that single target within the cell. In the past 2 decades, new in vitro models have been identified. And this is really important because in vitro models tend to be faster and less expensive than animal models. Importantly, a 3D tumor model was developed where the tumor cells grow into what looks very similar to an actual tumor, unlike traditional models where you have a petri dish and cells growing in a monolayer on the surface. In this 3D model, like I said, you have something that looks more like a tumor, and it's being recognized now that this dish, whether it be plastic or glass, really doesn't replicate the human body very well, mainly because it's so hard and physically hard, and that the interaction among these cells isn't entirely normal. It isn't recapitulating what's happening inside of a person when they have a tumor. One of the major steps in developing a new drug is determining the therapeutic index. And I know we've gone through this a couple of times before, but we wanted to make sure that you really understood what this was talking about. The therapeutic index is a comparison of the doses that would be toxic to cells other than the ones you're interested in treating, and the doses that would be therapeutic to the cells that you're interested in treating, either making better or killing in the case of cancer. So let's draw this out and go through a couple of different scenarios where you have, say, on the y-axis, percent effect. And, for us, effect is death, right? We want our Drosophila to die. On the x-axis, you would have increasing doses. So you have low doses down here and high doses up here. If you were to take whatever your model is, whether it be, let's just limit the discussion to chemotherapies. If you were to take, say, cancer cells or Drosophila as a model of a tumor, and you kept treating in different settings, say in different vials or in different culture dishes, many, many, many different doses, ideally what you're going to see is a curve that looks like this. And this curve, when it's 50% effective in the cells that you're interested in, say, we'll just call these cancer cells. At 50%, you have an effective dose that works on 50% of the subjects. And the way that you determine this is by, for each of the doses, graphing the percent effect. And then you connect the dots. In order to figure out the effective dose that works for 50% of the cells or 50% of the subjects, you figure it out based on the percent effect here, and then you draw a line down, and this here is the dose that is the ED50. Now I'm going to change colors here so that we can see something different, which is the TD50. Now ideally something that's toxic is going to be at a higher dose. So rather than doing cancer cells, maybe you would do something, you would test these same doses along the x-axis in something like liver cells, because we don't want to kill liver cells, we want to protect them. And I think what you would expect to see is that a much higher dose will start inevitably to affect cells that you're not interested in affecting. It's called off-target effects. And you do this exact same thing where you'd test your liver cells at the different doses, plot them as dots, and then connect the dots, figure out where 50% is, and here's your TD50 dose. Now, the reason that we do this is because we want these two doses to be really far apart. The farther apart they are, the less likely it is that you're going to choose a dose for a patient. Say, let's see. Say here. And if we go down, we're going to see, okay, that's great. But then we're hitting this curve here. And you might expect then about 10% of liver cells or 10% of patients would start to experience toxicity. Now, in some patients, you might have to give them a higher dose. Maybe they have a more advanced cancer. Maybe they are bigger. Bigger people need higher doses. And in that case, you draw the line down, and now you have a problem, because all of these people, in addition to the person who's being successfully treated for cancer, all of these cells are also going to be negatively affected. It's going to be toxic in perhaps 50% of the cells. So the farther apart these lines are, the less likely it is that you're going to have any overlap at all. Because even at the highest dose you would give somebody on this ED50 curve, you'd have very, very, very few patients who are experiencing toxicities. So you want it to be really far apart. Okay, in the Discovery Lab, as you know, we are interested in finding new chemotherapies that work as a combinatorial therapy with radiation. And the way that you've been doing this is you've been using third instar larvae, you've been irradiating them, and immediately putting them into a vial that contains compounds, individual compounds. We then allow them to eat the food, climb the wall, form pupae, and then we ask who survives. The ideal case is we get 0% survival. This is what we want, because we want these little guys here to die, because they're our cancer model. If we irradiate only and don't add compound, we expect these larvae to close at a rate of 50%. And this is an important consideration for your own work. When you propose your independent study project, or when you're writing about it, you want to keep this in mind, that if you're proposing radiation, the dose that you pick for radiation is going to be expected to kill 50% of the larvae. And this gives you enough room to see how much more effect the compound has. Now, some of you might be proposing to use mutant flies. You don't have to do this, but using the grapes gene mutation in flies for your independent project will help bring your project a little bit closer to what's actually seen in patients. And this is actually of great interest to Tintin Sue's lab. So I think we would recommend that you include grapes mutations in your independent project, but it's not required. So just to be clear, the way that this works is the grapes gene is basically monitoring for DNA damage. So the way that I like to think about this is, if this is DNA, in a totally normal case where the cell is dividing and the DNA looks really good, you have a few proteins sort of just hanging around doing their jobs. One of them is called ATR. Another, we're going to use the grapes human homolog, checkpoint kinase 1. And another one is called CDC25. And these proteins are all just hanging out because, like I said, the DNA is doing its thing. This is a kinase, so it adds phosphate groups. This is a kinase. It also adds phosphate groups. And this is a phosphatase. So it removes phosphate groups. And when everything is normal in the cell, CDC25 is an active phosphatase. In other words, it's actively removing phosphate groups from a complex called CDK cyclin. And this normally has a phosphate group stuck to it, but CDC25 is able to remove that, so now CDK and cyclin are just hanging out by themselves. When they're not phosphorylated, this allows mitosis to occur. So this is the normal state. Checkpoint kinase 1 is not doing anything. ATR, which is also a kinase, is not doing anything. CDC25, which is a phosphatase, is active. It's dephosphorylating this complex here, and it's allowing mitosis to occur. So what happens when there's DNA damage? And this is a totally normal situation. There's nothing weird about this. We're not talking about mutants yet. This is just the normal case of a cell dividing and replicating its DNA. So say there's a single or double strand break. This change in shape of the DNA allows ATR to get in there and associate with the DNA. It changes its shape so that the kinase becomes active, and it is able to add phosphate groups to things that have the right shape. Checkpoint kinase 1 has the right shape. It phosphorylates it, and that phosphorylation allows this kinase to become active. So it floats around and it looks for something that's the right shape, and it allows it to add the phosphate group. And in this case, it's adding the phosphate group to CDC25. Now, when CDC25 is phosphorylated, this change in shape that's produced by the addition of that phosphate group tags it so that other proteins in the cell recognize it and degrade it. Now, without CDC25, this doesn't happen. And what happens here is that instead of progressing to mitosis, you have cell cycle arrest. And again, in a normal cell, this is perfectly normal. This is a good thing for the cell. You want this cell to stop what it's doing, don't divide if there's damage to the DNA. And this gives the cell time to repair. Now, you can go through many iterations of what if ATR is gone? What if checkpoint kinase is gone? And all of these things, if they're not working properly, you're not going to get mitosis to occur properly. Now, we use flies that harbor a Grapes mutation because Grapes mutations are really common in head and neck cancers, and that's the type of cancer that we're modeling. And people ask, well, what are head and neck cancers? They're literally any cancers that occur in the head or the neck. So things like thyroid cancer, glioblastoma, cancer that come from neurons, throat cancers, cancers in the sinuses, that happens sometimes. So any cancer, any tumorous cancer that occurs in the head or the neck is something that might have mutations in checkpoint kinase 1. Now, what's interesting is you might think, well, it's probably that the mutation causes it to go away. But in fact, many of the mutations cause the checkpoint kinase 1 to work better. And the way that that's beneficial to a cancer cell is that if checkpoint kinase 1 is working better, it's better able to detect damage in a cell where damage is occurring at a higher rate because cell division is occurring at a higher rate. So this is beneficial to the cancer cell. Now, unfortunately for us, the Grapes gene is a maternal effects gene. And what that means, very basically, is that in a maternal effects gene, the mother has to have a normal copy of the gene. because these genes, grapes isn't the only one, there are other ones. These genes are required from mom because they're necessary for the development of the embryo at a time when the embryo isn't making its own genes. So mom needs to put it into the embryo while these nuclei are dividing and before the nuclei are making their own genes. If mom doesn't have a normal copy, this embryo dies. And this is a little bit of a bummer for us, because if we had flies that we were trying to breed, we want them to be minus over minus, meaning that they're nulls. We don't want the flies to have the grapes gene, but we can't do this. We can't cross a null with a null. And I'm not talking about any balancer chromosomes yet. You can't do this because mom is never going to have a normal protein to give to her offspring, and they're going to have all dead babies. Not good for us if we want to maintain an enormous line of flies as we have in Discovery Lab. So then you might say, well, what if you did a breeding like this? A het mom with a het dad. And then you do get a certain percentage. You get 25% that are nulls, 50% that are hets, and 25% that are wild types. And this isn't so bad. Yeah, you would lose 75% of all of the offspring because they wouldn't be of interest to us. But there are a couple of problems here. One is that we can't tell the difference between this fly, and this fly. They all visually look identical. So to be able to tell the difference is impossible. But we're banking on being able to use these guys. Another problem is that the wild types breed a lot better than the nulls do. So you could have a null male go on and produce offspring. That's not a problem. But these offspring they're not going to do as good of a job at identifying DNA damage as the embryo is developing. So these embryos are going to suffer a little bit. And over time, what you see statistically is that the wild types just take over the whole colony. So the number of nulls that you get decreases over time. This is definitely not ideal for us in a lab where we're already breeding tens of thousands of flies, when we're going to lose 75% of them, and eventually we're going to lose all of them. So the way we get around that is by using a balancer chromosome. In Discovery Lab, we use balancer chromosome number 2, and it has a normal grapes gene on it that's connected to curly wings, and it's also connected to a protein called GFP, green fluorescent protein, where if you shine a blue light onto those flies, they glow green. So these 3 genes travel as a unit. The existence of the balancer chromosome allows us to do a couple of things. One is we're going to eliminate the wild types, because any fly that has 2 balancer chromosomes is dead. So this also allows us to maintain breedings that are generally het-het breedings, so that we maintain a stable of at least 25% of the population being nulls. And this number does not decrease. It stays stable over time. Finally, we can see the genotypes, because any fly that has a wild type copy is also going to glow green and have curly wings. We're interested in nulls. And because the mutated version is on the normal chromosome, these flies are not going to glow green, and they're not going to have curly wings. So we can see that they appear to be normal flies. These are the ones that we're interested in. So in your final research paper, you don't need to go into a great deal of detail describing this. And if you're not using the grapes, or you're not proposing to use the grapes mutant flies, then you don't need to discuss this at all. You'd be using wild types like we have in the lab. But what you do need to know is if you propose using the grapes genes and we give you data, you need to be able to interpret those data. And in that case, it's going to be really important that you understand this concept of balancer chromosomes, also of maternal effects, and of the Grapes gene and what it does. Okay, so let's move on and do a brief overview of clinical trials just to highlight the important things from the reading. And so hopefully this will serve as a review for you for your final exam, or the third exam, which is cumulative. You already read about this, but clinical trials occur in generally 4 phases. The thing I want to point out here is that the first 3 phases use an increasing number of people. So they have an increasing number of subjects or volunteers. And after phase 3, you get FDA approval. This is when you'll start to see this drug in the pharmacies. It occurs before phase 4. Phase 4 occurs after FDA approval, which is not fully appreciated by a lot of people. So when a new drug comes on the market, that drug is technically still in clinical trials. And what they're doing is they're monitoring for long-term effects, or effects that they maybe weren't able to see in a larger population of phase 3, because phase 4 is going to have the most number of subjects because now it's available to everybody. Clinical trials are hard to get to, hard to perform, and massively expensive. So they're running into the billions of dollars now. We are down here. So Discovery Lab is sort of at the 10,000 compound phase, although these 10,000 compounds imply that they're completely random compounds. The compounds that you've been testing, we know something about. We know their structures. We know that they have at least some promise as chemotherapies. So you are somewhere between this blue segment and the green segment. We're preclinical, and at 250 compounds, this is where you start doing animal testing. So you start out with 10,000 compounds. Many of those, most of them, are going to fail. They're not going to work. And then after testing all 10,000, you'll end up with maybe a set of 250, and you're going to keep testing those, and a small number of those are going to get tested in animals. Once they are successfully tested in animals, they may go to clinical trials. So say you were an enormous company and you were able to start 5 clinical trials at the same time. From your original 10,000 compounds, you have 5 because every other compound has failed. I should point out here too, I think that we will talk about this when we talk about bioethics, but the FDA does not require animal trials. This is something that I think is a misconception. Drug testing does not require testing on animals, but the caveat is it's extremely difficult to get the information that's required of the FDA from anything but animals. So just so you're clear on where your drugs are coming from, I would say almost all of them were tested in animals at some point. So after those clinical trials, you may end up with one drug that becomes FDA approved and is a drug that can be prescribed by a doctor. So when you go into this field and you're interested in testing drugs to identify new chemotherapies or new drugs to treat diseases, your odds of developing a drug that will actually go into a patient are at best 1 in 10,000. The amount of time from where we are until, say, one of your drugs becomes FDA approved is a long time. So this is probably somewhere in the range of, let's just say we're 4 years out of clinical trials in the absolute best case. So, you're talking about 12 to 13 years, which is a little bit disheartening. Although there are ways to make this go faster through the new phase 0 microdosing studies, which are intended to identify problems with the drug before they even go into phase 1. But most drugs are failing in phase 3. The goals of the clinical trials, we already know a lot about drugs going into clinical trials, but there are lots of different types of clinical trials. the ones that we've been focusing on are treatment trials, but there are also clinical trials that involve diagnostic tests, preventative treatments, and also devices. So devices like pacemakers, for example, if there was a new type of pacemaker, it would go through clinical trials slightly differently. The requirements are a little bit different, but it still needs to be monitored by the FDA and get approved in the same kind of way as a drug does. So this is another way of looking at the clinical trial process. As I said, there's a preclinical phase, and this is us, the Drug Discovery Lab. The first 3 phases that involve a fairly small number of patients. I know if you can imagine testing in 3,000 patients, it seems like an awful lot, but in the grand scheme of things, it's not. FDA approval may occur after phase 3, before phase 4, when it becomes available to the general public. Now, the important thing here in phase 1 is this is a very small number of patients. Can be as little as 10 patients. And by patients, I really mean volunteers. These are healthy people. And to be honest, these are young, healthy people who are paid. And you can probably guess who these young, healthy people are. These tend to be college students and people who are financially disadvantaged. So I'm gonna write poor people here, because these are the people who are most likely to be willing to take a drug for, say, $500 to see if it's going to be safe. We already know that these drugs are safe in animals, but we really don't know if they're safe in patients. So they do tend to pay a good amount of money to people to participate in this phase of the trial. Now, once the phase 1 is completed so that they know it's safe, then they ask, does it work? And this is tricky because you don't want to do this in patients who have real hope of being treated with something that already works, right? So the first misconception is that there are no placebos generally in this phase. It's generally not ethical if there's another drug on the market that is remotely effective to give someone a sugar pill or your new drug. You have to give what's called the gold standard treatment or the new drug. So they won't be any worse off than they would have if they had just gone to their doctor. Now, you might ask then, who are these people? These people are sick. These are very sick people. Because who are the people who are likely to say, oh, I know that there is a gold standard treatment on the market, but why don't you just give me the new drug? Let's give it a shot and see if it works. For people who have illnesses that will kill them, it's very unlikely that they're going to try a new drug unless they're extremely sick. So in this second phase, you're asking, does it work? But you're asking it in a population of people that's really tricky to treat and really tricky to interpret, because these people are taking tons of medications and they have a lot of other issues going on besides just the issue that you're willing to treat, that you're wanting to treat. So in the third phase, this is when you start dealing with sort of a more normal population of people who have the disease. But it's small. You're starting to see some interactions with other drugs. So for example, people who have cancer are often taking blood pressure medications. They're also often taking pain medications. So you start to see interactions with other drugs among these couple of thousands of patients that you weren't really able to see in phase 2 because these patients are just too complicated, and you definitely weren't able to see in phase 1 because these patients are healthy. They're not taking anything else. This is a super clean study where you just want to see if it's going to kill them. So these patients in phase 3 are looking a little bit more normal, more like the typical patient, but it's still a rather small number of patients. It isn't until you get to phase 4 where it's been FDA approved that you just start to see things that you haven't seen before ever. So not surprisingly, a lot of compounds, even though they make it through preclinical testing, phase 1, phase 2, fail at a very high cost in phase 3 and phase 4. And we're talking about hundreds of millions of dollars invested in drugs that have made it all the way through to phase 3, but don't get FDA approved because suddenly somebody starts dropping dead because of an interaction with another secondary illness that they've developed. So in this lecture, we have covered our business items and upcoming due dates. We covered the fact that there's nothing happening in the lab and that the vast majority of the flies that we had in the lab have been eliminated. We did a thorough review of what we've covered so far so that when we move forward to the next 2 units, you can focus on those and studying for the cumulative exam, which will take place in 2 weeks. We also talked about drug discovery development, specifically clinical trials.